Hi, dear children. Welcome to the next session on the chapter Respiration in Plants. Dear children, in the last module, we learned that the pyruvic acid, in the first step of its oxidation, it gets converted to acetyl coenzyme A, isn't it? Acetyl coenzyme A in the matrix. And how many carbon atoms are present in pyruvic acid? We know it is 3 carbon atoms. And during this process, there is a decarboxylation taking place. There is one molecule of carbon dioxide is removed. That means from pyruvic acid, one carbon is removed. So what will be the remaining carbon atoms in the product in the acetyl coenzyme A? When carbon dioxide is released out, it will have only 2 carbon atoms. Okay, and this acetyl coenzyme A having 2 carbon atoms enters into tricarboxylic acid cycle. Let us learn about the TCA cycle. The TCA cycle is always called as the Krebs cycle. Why it is called as Krebs cycle? Because it was first elucidated by Hans Krebs. It is also called as citric acid cycle. Why it is called a citric acid cycle? Because the first compound formed as a result of this cycle is citric acid. And since it is a tricarboxylic acid, it's also called as tricarboxylic acid cycle. So here we can just once again revise what we have learned. The pyruvic acid with 3 carbon atom undergoes oxidative decarboxylation in the presence of NAD+, that is coenzyme NAD+, and coenzyme A in the presence of enzyme pyruvate dehydrogenase and magnesium ions. It gets converted to acetyl coenzyme A, and here one molecule of carbon dioxide is released with the reduction of NAD+ to NADH+ H+. Now this acetyl coenzyme A, what happens to it? It undergoes condensation. Okay, condensation of acetyl group of acetyl coenzyme A with the oxaloacetic acid. Oxaloacetic acid is having 4 carbon atom and acetyl coenzyme A is having 2 carbon atom. So condensation of the acetyl group takes place with oxaloacetic acid abbreviated as OAE and water. In the presence of water, it gets converted to form citric acid, and this process of condensation or conversion of condensation of oxaloacetic acid with the acetyl coenzyme A is mediated or catalyzed by the enzyme called as citrate synthetase. Okay, and during this process, the coenzyme A is released out. Okay, and citric acid, that is the first stable compound, is formed. Hence the cycle is called as citric acid cycle. And this citrate formed, it gets isomerized to form isocitrate. Okay, the citric acid gets isomerized to form isocitrate, and this isocitrate, it undergoes decarboxylation. What do you mean decarboxylation? It is the removal of carbon dioxide or release of carbon dioxide. So carbon dioxide is released and this alpha ketoglutaric acid is formed from isocitrate. Okay, so decarboxylation takes place and citric acid, which has been isomerized to isocitrate, gets converted to alpha ketoglutaric acid, and in this process NAD+, the coenzyme NAD+, is reduced to NADH plus H plus. And again, this alpha ketoglutaric acid. Okay, now let me once again get you a point. See how many carbon atoms are there in citric acid? 6 carbon atom. And again, decarboxylation has taken place. That means 1 carbon has been removed. So what will be the number of carbon atoms in alpha ketoglutaric acid? It is 5 carbon atom. Okay, so this 5 carbon atom alpha ketoglutaric acid, it undergoes the next step of decarboxylation. Okay, and during this process, what happens? It gets combined with a coenzyme A. Okay, to form succinyl CoA. Okay, so decarboxylation of alpha ketoglutaric acid takes place to form succinyl coenzyme A in the presence of coenzyme A. And succinyl coenzyme A, again, it is having 4 carbon atom because 1 carbon atom has been removed over here. Due to the release of 1 carbon atom, the 5 carbon alpha ketoglutaric acid is being now converted to succinyl coenzyme A in the presence of coenzyme A. And this succinyl coenzyme A is converted to succinic acid, which is a 4 carbon atom. Okay, and here the substrate level phosphorylation takes place. Substrate level phosphorylation means a phosphate group has been transferred from the succinyl coenzyme A to GDP, an energy-rich compound called as GDP. GDP stands for guanosine dinucleotide. Okay, guanosine dinucleotide. So this guanosine dinucleotide, once it receives a phosphate, substrate level phosphorylation takes place, and a GTP, an energy-rich compound, GTP is formed. GTP stands for guanosine triphosphate. It is an energy-rich compound just like ATP. And it is a coupled reaction. That is, the GTP immediately it transfers its phosphate group to ADP, and thus GTP gets converted to GDP, and the ADP which has received the phosphate groups get converted to ATP. So here, when the succinyl coenzyme A is converted to succinic acid, there is a substrate level phosphorylation. As a result, one molecule of ATP is being synthesized, and it is also coupled with the reduction of NAD plus to NADH plus H plus. The succinic acid, the 4-carbon atom succinic acid, gets oxidized to fumarate or fumaric acid and then to malic acid. And during this process, FAD, a coenzyme FAD, is reduced to FADH2. FAD stands for flavin adenine dinucleotide. So FAD, flavin adenine dinucleotide, gets reduced to FADH2, and subsequently the fumaric acid, succinic acid, which is converted to fumaric acid, and fumaric acid ultimately gets converted to malic acid. And the last step, the malic acid gets oxidized to oxaloacetic acid, and during this process, the NAD plus, the coenzyme NAD plus, gets reduced to NADH plus H plus. So thus, oxaloacetic acid is regenerated. So once again, let us recollect what we have studied. The acetyl coenzyme A, which enters into the citric acid cycle, gets condensed with oxaloacetic acid in the presence of water with the release of coenzyme A to form a 6-carbon atom, 4 carbon in oxaloacetic acid and 2 carbon in acetyl coenzyme A. And the coenzyme A is released and it forms citric acid. This citric acid gets, and this reaction is catalyzed by citrate synthetase. The citric acid undergoes isomerization to form isocitrate, and this isocitrate, it undergoes the decarboxylation with along with the reduction of NAD plus to NADH plus H plus to form a 5-carbon atom, alpha ketoglutaric acid, with the release of 1 carbon dioxide. And this alpha ketoglutaric acid, it combines with coenzyme A to form a 4-carbon atom molecule compound, a 4-carbon atom compound called as succinyl coenzyme A. And over here also there is a decarboxylation takes place, and thus the 5-carbon atom is converted to 4-carbon atom succinyl coenzyme A. And this succinyl coenzyme A, it is converted to succinic acid with substrate level phosphorylation and with the synthesis of GTP, and this GTP again gets converted to GDP with the transfer of phosphate group to ADP, formation of ATP. And this succinic acid gets oxidized to fumarate and then to malic acid with the reduction of FAD to FADH2. And this malic acid again gets oxidized to oxaloacetic acid with the reduction of NAD to NADH plus H plus. So at 3 points of this TCA cycle, tricarboxylic acid cycle, NAD was reduced to NADH, which all points when isocitrate was converted to alpha ketoglutaric acid, when alpha ketoglutaric acid after combining with succinyl coenzyme A, the succinyl coenzyme A is converted to succinic acid, and also when malic acid is regenerated to oxaloacetic acid. And at one point, FAD is reduced to FADH2. And for this tricarboxylic acid cycle to continue, continuous regeneration of oxaloacetic acid along with formation of NAD and FAD has to take place for the tricarboxylic acid cycle to continue. Okay, so it also requires regeneration of NAD and FAD from NADH plus FADH2. Okay, now let us summarize the equation of Krebs cycle. The pyruvic acid plus 4 coenzyme NAD plus 2 molecules of water plus 1 molecule of ADP plus inorganic phosphate plus FAD. Okay, so pyruvic acid, as we know, it is an end product of glycolysis. 4 NAD, 1, 2, 3, and the fourth one is formed when, that is fourth one is formed when the pyruvate gets converted to acetyl coenzyme A. This NAD is needed over here, over in this reaction, where NAD is reduced to NADH plus H plus. And ADP, here there's a substrate level phosphorylation taking place when the GTP transfers its phosphate to ADP and it gets synthesized to ATP, plus inorganic phosphate and FAD. Okay, FAD is not written over here. There is FAD also. And this is taking place in the mitochondrial matrix, which is 3 molecules of carbon dioxide. 1, 2, and 3 over here. Okay, so 1, 2, and 3. And 4 NADH is formed. 1, 2, 3, and 4 over here. And in addition to it, 4H plus along with NADH and FADH2 over here, plus 1 molecule of ATP. And as we know, glucose, when it was broken down in glycolysis to pyruvic acid, 2 molecules of pyruvic acid was formed. So we can double all the amount, all the products formed over here. So that is 6 carbon dioxide plus 8 NADH plus 8 H plus 2 FADH2 plus 2 ATP. So that is the net gain of products formed after a Krebs cycle. Okay. So dear children, hope the concept is clear for you. Thank you.